This tutorial focuses on separation techniques. The first aim is to describe the difference between miscible and immiscible substances, then explain how to carry out 3 separation techniques, and then explain the importance of RF values. That's related to the separation technique of chromatography. Now obviously our planet is filled with useful materials, but very seldom are they just there alone for us to take as if they were an apple off a tree. Normally they're combined to other things or mixed in with other things. I mean, for example, take salt in seawater. That is mixed in the water. We have to apply a separation technique to try and separate the salt from the water. But sometimes separation techniques are needed when there are disasters, such as an oil spill, where the oil clings to all sorts of sea life, as well as rocks, as well as obviously it floats on top of the water, so we need to find a way to separate it. So to start off with, let's define 2 important words needed for this topic: miscible and immiscible. Basically, miscible substances mix well. For example, here I've got some water. I'm going to add some squash and mix it around. And you can see that it's mixed nicely with the water. Immiscible substances do not mix well. For example, here I'm pouring some oil into some water and you can see it separates out, leaving a layer on top. Oil is less dense than water, so it floats on top, but you can see they're very much separated, not mixed in. So oil and water are immiscible substances, but squash and water are miscible substances. So that is how you describe the difference between miscible and immiscible substances. Aim 1 done. So now we're going to look at 3 different separation techniques. The first one is use of a separating funnel to separate immiscible substances such as oil and water. So I've dyed some water here, as you can see, and there's oil which is less dense floating on top. This piece of equipment is called a separating funnel, and it has a little tap here you can open and close. So the method is simple. Open the tap, drain off the most dense liquid into a beaker, so water is more dense, so it'll fall through this tube first, and then close the tap once it's all drained off. You can see this in action now.
So you have a separating funnel, and because oil is less dense than water, it sits on top. It's lighter, it stays on top. So that means you can just use a separating funnel to drain off the more dense substance by turning the tap, and it'll start to come out. Now you've got to do it slowly when it gets towards the end, like now. Drip, 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 drip. Just a little bit. And then you see that now it's separated.
So the key point here is a separating funnel separates substances according to their density, and they must be immiscible, substances that don't mix, such as oil and water. We can use fractional distillation for miscible liquids, and here we separate things according to their boiling point. For this you need distillation equipment like this simple setup here, or a more complex fractional distillation column, as you may remember in the tutorial on separating crude oil. So in this setup I've got water and alcohol in here. They have different boiling points, you see. Water boils at 100 degrees Celsius, but alcohol at 70 degrees Celsius. That's why chefs commonly put alcohol in food, such as wine, with no fear that the alcohol will stay in the food because it boils off before the water does, leaving just the flavour. So the idea is we're going to heat this up to 70 degrees, but not 100 degrees, because at 70 degrees the alcohol boils, but the water doesn't. So as long as it's below 100 and above 70, we've got no problem. So the alcohol turns into a gas and is passed down this tube here. This tube is surrounded by a water jacket, another layer of tubing where cold water is pumped through it. That cools down the gas as it travels through, forming a liquid form of alcohol. So it condenses and then drips out here. So we get pure alcohol at this point. You can see this now. So you can see it boiling away over here. So you can see the water and alcohol boiling here. The alcohol boils off, condenses here where it's cooler, and then drips out over here. And that's all because the water cools down the alcohol, turning it from a gas back into a liquid. In exams, it may ask you how you perform the fractional distillation of air, because air is a mixture of different gases, as you can see here. This is how it works. We determine the modern composition of our atmosphere, the modern balance of gases in our atmosphere, by using the process of fractional distillation. This is when we separate substances according to their different boiling points. To do this for air, we had to filter the air to get rid of any dust, which we didn't need. Then we cooled the air down to minus 200 degrees Celsius. As you start to cool it down, the water vapour condenses first and becomes a liquid. So that's how we remove water vapour. Next, the carbon dioxide in the air freezes and becomes a solid. So that's how we remove the carbon dioxide. What remains is nitrogen and oxygen largely, and a bit of argon. So we've cooled these down to minus 200 degrees Celsius where they're both liquids.
We then slowly heat them up so these gases enter the fractionating column. A fractionating column is a large chamber where at the top there's a cooler temperature and a warmer temperature at the bottom. So nitrogen and oxygen enter. Due to their separate boiling points and the fact there's a temperature gradient in this fractionating column, they basically separate out, so nitrogen will come off here and oxygen here.
So you can use fractional distillation to separate miscible substances, liquids or gases, and it separates them according to their boiling point. We can use chromatography, and this is the third separation technique you need to know, to separate miscible substances and identify them. Usually something like a mixture of dyes in an ink or mixture of chemicals in a fruit juice or something like that. Firstly, you must get some filter paper in a strip like this. About 1 centimetre up from the filter paper, you draw a pencil line. This is called the baseline. It must be pencil and not ink. You'll see why later, but ink will dissolve. We want this mark to stay there. Next, you have to prepare the dye you want to separate out into its different chemicals. So here I'm grinding a leaf because a leaf contains chlorophyll, which is the green pigment in leaf, but actually it's a mixture of different pigments. I'm using a solvent, propanol, to actually dissolve the chlorophyll in different pigments and form a little juice which I can then extract. Once the juice is concentrated enough and dark green, then I can spot the dye on a pencil baseline on the filter paper. So look, there's my pencil baseline and I'm spotting it. I basically spot it, then let it dry, then spot it again, then let it dry. I want it to be as concentrated as possible without spreading too much. Next, you place in a beaker or a boiling tube containing a solvent. So you use a special solvent which will dissolve this stuff, these chemicals. And what will happen is the solvent, which must be below the pencil line, can't start above it, it must be below, will start to soak up the filter paper. As it soaks up, it will dissolve all the chemicals that make up this dye and it will transport them up the paper. Now, according to the mass of the individual chemicals that make up the dye and the solubility, how readily they dissolve in the solvent, different marks will be made up the paper. The chemicals which have the greatest mass will be deposited earlier on in the filter paper, and those which have the lightest mass will be transported further up because you need less energy to transport them, to carry them. Just like if I asked you to carry a heavy box or a light box, you'd be able to carry the light one further. So you can see that now. You can see it's running up using time lapse. It speeds it up a bit. So as the solvent travels up the filter paper, different chemicals in the mixture form separate spots on the paper. Sometimes it can get a bit blurry and messy, but you can see there's two different colours, two different pigments here, dark green and sort of yellowy green one here. That is how you explain how to carry out the three separation techniques, being using a separating funnel for immiscible substances, using fractional distillation for miscible substances, and also using chromatography for miscible substances. Now, chromatography doesn't come up every year in the exam, but when it does, it's often a part which students neglected to revise. So when it comes up, it can really hit students hard if they haven't looked it or just basically overlooked it in their revision. What makes it more frustrating for me as a teacher when I mark students' exams is just knowing how easy it was and how many easy marks students threw away by not covering it. So how do we interpret chromatograms? Well, here's a completed chromatogram. This is the filter paper with different chemicals being deposited at different positions. You can see the solvent, the liquid that was running up, also finishes at this point. This is called the solvent front. You can see our pencil line where we originally marked over there, staying in the same position. And the chemicals with the larger mass are here, and the chemicals with the lowest mass are here. Now let's say that was an unknown chemical and we wanted to identify what it was. We could compare this chromatogram to other known chemicals and help us identify it. So here are the kind of questions you might be asked. Can you firstly identify the unknown chemical? Well, if you look at the pattern, there's a yellow spot here and only this one has a yellow spot here, a green spot here and only this one has a green spot here, and a red spot here. Now they all have red spots here, but only this one has green and yellow. So it must be chemical B from the known chemicals. Secondly, which chemical is present in all chromatograms? Well, you can see that X, the red chemical here, is present in all three because we have the same coloured dot in the same position, so that's really easy. Next, which chemical in the known chromatograms is not present in the unknown chemicals? So which present in here but not here? Well, you can see it's W here because we've got a blue spot here in this position and there's no blue spot here. Hopefully you can see how easy it is, and also why it's frustrating when I see students can't answer it because it's such a simple thing to answer. Every chemical substance that can be separated in such a way is given an RF value. Now, RF values are a ratio, and they tell us how far the chemical has moved in relation to the solvent. Very simple to work out. So let's say I wanted to work out the RF value of the yellow pigment here. I would measure with my ruler, let's say in centimeters or millimeters, but whatever you do, use the same units. I've used centimeters. I measure from the pencil baseline to the tip, the leading tip of the pigment, and let's say it measures out as 8 centimeters. Then I do the same, but for the solvent front, from the pencil baseline all the way to how far the solvent transported itself. And to work out the RF value is just the distance travelled by the chemical divided by the distance travelled by the solvent. So 8 over 10, which is 0.8. If you had an RF value of 1, that would mean that the pigment's actually transported right up to the edge of the solvent front. You will never ever get an RF value over 1, because that would imply that the pigment has leapfrogged over the solvent front and somehow magically appeared above it. So RF values are recorded in tables which are used by scientists. Two uses are in the food industry to identify unknown food additives and forensic science to identify drugs. And that is how you explain the importance of RF values.